What are ribonucleases? Ribonuclease, or RNase for short, is a class of enzymes that turn the chains of nucleotides and nucleic acids into smaller units. Obviously, ribonuclease catalyzes the degradation of RNA molecules and can be found in all living cells, both prokaryotic and eukaryotic. There are two broad groups of RNases, endoribonucleases and exoribonucleases. Both of these differently cleave various RNA species doing the same job differently. RNA degradation is as ancient as it is important. The cellular concentration of a given transcript messenger RNA depends on its synthesis and degradation. This essentially controls the levels of each protein in the cell. RNases are also the first defense against RNA viruses. The extraction of RNA in molecular biology experiments is made quite challenging as the RNases degrade RNA molecules into smaller constituents. RNases generally have a very high specific activity towards their substrate, RNA, which means even the smallest amount of RNase can interfere with the sample of RNA. It works on the phosphodiester backbone of ribonucleic acids and is involved in the transfer of a phosphate group from the 5' position of one nucleotide to the 3' position of the next nucleotide in the chain, and plays a key role in the maturation of messenger and non-coding RNA, and is also responsible for ridding cells of obsolete RNA. Studies have shown that a cytotoxic effect, the cleavage in the RNA molecule leading to the inhibition of protein synthesis, and including apoptosis, is brought about when RNase is applied to the outside of a cell, and that this cytotoxicity increases 1,000-fold when artificially introduced into the cytosol.
Ribonucleases are everywhere. RNases are very stable and difficult to inactivate. To ensure success, it is important to maintain an RNase-free environment, starting with RNA purification and continuing through analysis. Sources of unwanted ribonuclease in a lab is mostly attributed to microorganisms, but can also be found on humans in shed hair, flaked skin, and fluids such as tears, saliva, mucus, and perspiration as an antimicrobial defense measure. It is a true pest in a molecular lab when studying RNA, and measures must be taken to keep samples uncontaminated. Wearing lab coats, regularly changing gloves, and avoiding turbulent airflow and particulates. Use sterile techniques when handling the reagents used for RNA isolation or analysis. Ribonucleases are very robust molecules, capable of withstanding extreme environments such as mammalian guts. The isolation of one type of RNase, bovine pancreatic ribonuclease A, involves treating it with sulfuric acid and boiling, both of which it survives. Because of this stability, autoclaving cannot be assumed to make implements RNase-free. RNases rely on histidine residues in their active site for catalysis, so diethyl pyrocarbonate, or DEPC, which is a histidine-specific alkylating agent, can inactivate them. Autoclaving will then remove the DEPC. Their activity is almost decimated by autoclaving, but a small portion remains. While autoclaving partially destroys RNases, there is no hope of them withstanding dry heat. Dry heat oxidizes RNases at 180 degrees Celsius for 2 hours.